everyone. Sorry for being late. Yeah, thanks for this opportunity to present here today. I'll be talking about uranium-thorium and uranium-lead dating, giving a bit of an overview of these two chronometers and where I think we're positioned in the field. And then I'll give two examples of applications for uranium-thorium and uranium-lead dating. Yeah, so let's get started. So I'm currently based at the University of Cape Town, just for another 2 months, then I'll move to the Berkeley Geochronology Center. But my background is very much in isotope geochemistry, specifically uranium-thorium and uranium-lead dating. So this is where I sit most comfortably, but a lot of the applications of why we do the dating is for paleoclimatology, and then to some extent also the implications that has for human evolution. I also work on other fossil sites, some not associated with hominin fossils, but faunal fossils, and we also look at fossil pollen in speleothems. So there's a bit of evolution in a larger context than just human evolution. Yeah, so where these three intersect is where I'm quite happy. And yeah, the way my talk is organized is indeed first we'll talk a bit about the applications or about the chronometers themselves, and then look at the more uranium-thorium realm and some proxy data from there, and then proxy data over the last 3 million years where a lot of the chronology comes from uranium-lead dating. So first of all, why are we using speleothems? We already had a bit of an introduction, but speleothems are arguably one of the best terrestrial archives. So for a number of reasons, but some of the most important ones is that they occur globally. So everywhere you find karst, limestone and dolomite rocks, you often find caves that have developed in these limestone host rocks. So that allows for very nice global comparisons and is a very good terrestrial, provides very good terrestrial proxies that can sort of complement what we know from marine records and ice core records, and is really to understand what's actually happening on the continents from terrestrial proxies. So there's multiple proxies that you can study through speleothems, so stable isotopes, the fluid inclusions, but to some extent, if it works, you can also look at pollen. And I'll show you an example today of how we're actually using the chronological data to say something about wetter and drier times. And yeah, probably the best advantage of speleothems is that we can date them really well. So we can produce really accurate and precise chronologies using uranium-thorium and uranium-lead chronometers. And I just plotted this here. This is from when speleothem research sort of kicked off in the 1960s. And I think because people understood that this is such a nice archive to complement marine and ice core records, that really proliferated the research in speleothem science. So uranium-thorium and uranium-lead dating is based on the natural radioactive decay of the parent isotope uranium into daughter isotope thorium, and ultimately into a stable isotope of lead. And this is just one of the 3 naturally occurring decay chains that ultimately decay into stable isotopes of lead. Probably also the most important decay chain for this chronometer. And we can use this for speleothems so well because speleothems act as a closed system towards these elements generally. That's because in the hydrocycle, these elements are strongly separated from each other due to different chemical behavior. So uranium is highly soluble in water, whereas thorium and lead are not. So they like to stay absorbed onto clay minerals and detrital material and are thus generally removed from the hydrocycle. The uranium sits in the water. So when that drips through the karst and ultimately enters a cave, then in an ideal world, when you have a nice stalagmite like this, only uranium is co-precipitated with the calcium carbonate when the speleothem forms. So from then on, the uranium is locked in and the clock starts ticking. So over time, uranium decays into thorium, and ultimately that will then over time decay into stable isotope of lead. And clever people have worked out at what rate these elements decay. This is what we call the half-life. And we can measure, then the half-life means the time it takes for half of the elements to decay away. We can measure the uranium, thorium, and lead isotopes with the mass spectrometer. So with all these knowns, then we can calculate an age. And for uranium, thorium, the limit is about 500 to 600,000 years, depending on your samples and your acquisition of your chemical protocols and your mass spectrometry. It depends whether you can push it more towards the 600,000 years. Anything older than that can potentially be dated with uranium lead. Actually, you can also date younger stuff with uranium lead, but uranium lead is quite laboratory intensive and a bit expensive, so generally things younger than 500,000 years are measured with uranium, thorium. And I just want to show this maybe a bit intimidating plot, but here you have the different isotopic ratios, the activity ratios, just to show you why we have this inherent age limit, or like limit of the chronometer for uranium, thorium. And that's because this is a disequilibrium dating method. So it's based on a disequilibrium between uranium and thorium. And after about 6 half-lives, this will approach secular equilibrium again. So you can see over here we have these isochrons, and when we sit at that limit is when they're too close to each other and we can't really well distinguish where we're at in age. So that defines the limit of this chronometer. But then for uranium lead, we don't have this problem. We have other problems. So there's a bit more involved. We need to correct for cases when there is initial thorium, or when you're looking at uranium lead when there is initial lead. This can be quite a big problem, but you can monitor it for uranium, thorium, and then correct for the initial thorium. With uranium lead, this can really be a limitation on how well we can date our samples. But there's ways that you can still get a good age if you have a few of your samples that have high initial thorium, lead. Then we also need to take into account initial disequilibrium between another uranium isotopes that's part of this decay chain, and that's uranium-234. So this, if you don't take this into account, then you can get very overestimated or underestimated, depending on what your initial ratio was. You can underestimate your ages. So this is really important to take into account as well. But after that, you can produce really nice, reliable ages. And the most applied method of doing this dating technique is through wet chemistry, so isotope dilution dating, where we use what is called ion exchange column chemistry. So here you can see a setup that we had when I was still at the University of Melbourne. John Woodhead's really the one who pioneered this technique for uranium lead dating. It's a bit similar for uranium thorium, but there's a lot more involved for doing uranium lead. We need to be super clean because there's a lot of lead in the environment that then can contaminate these samples that are generally not super radiogenic. That's because we sit really at the younger end of this chronometer. So there's not that much uranium that we have in the samples, and then they're also not that old, so there's not that much ingrown lead. So we need to be in a super, super clean lab and really try to avoid contaminating our samples. But this column chemistry is based on, so when you dissolve your sample and you put this on these columns where you have some resin, then you can activate that resin with some kind of acid and the sample will stick to that. Then with other kinds of acids, you remove the matrix, and ultimately, again with different kinds of acids, you can then extract the elements you're interested in. So for uranium lead, we're looking at the uranium lead, and for uranium thorium, we collect that in one go together. So for uranium thorium, we have enough with just a single aliquot from a speleothem sample to produce an age. It takes about 2 days in the lab, and then a day of mass spectrometry if you measure uranium and thorium at the same time. For uranium lead, there's a little bit more involved. You need at least 3 aliquots because we're producing what we call an isochron. So this is the Terra-Wasserberg isochron, and you need multiple aliquots, so multiple measurements to define this isochron well. So this upper intersect is where this defines how much initial lead there was, and the lower intersect is where we find the age. It's simple set. So there's a bit more involved because we need more aliquots to do this, but often we also need more than just 3, so you need to do the chemistry again. The chemistry is at least 3 days, at least what we did at the University of Melbourne, and then at least for a single batch, 2 days of mass spectrometry because you measure uranium and lead on different days. You can't do that at the same time with multi-collector ICP-MS. So this is some of the setup we have. This is our PhD student Georgina Luthi from Kenya, who is learning everything about the technique, and Robin Pickering, who's an expert in doing this. And here we are very happy to get going with the uranium thorium at the University of Cape Town as well. So there's another way of doing this, and that's using in situ laser ablation dating. So you use a fancy laser like this, and you mount your samples in a sample holder, and you can then shoot lines along your sample. Again, this is also largely pioneered by John Woodhead at the University of Melbourne. And this is mostly also made possible by these new generations of mass spectrometers that are called high resolution ICP-MS, and they can actually measure the different elements on the same day. So it's a very rapid technique, and another nice thing that you can do with this is that you can actually scan a line along the growth axis of your speleothems to see where the uranium actually sits. Because I didn't say that, but in my previous slides, what we need to successfully date with uranium lead is generally above 1 ppm of uranium and a few hundreds of ppb lead that is radiogenic. And speleothems generally don't have that much uranium, so if we're sitting at the younger end of these speleothems for the uranium lead dating, then you really need to know where you sample from. So with laser ablation, you can do a sort of reconnaissance to see, this is for 238 uranium and 208 lead. 208 is how we monitor the common initial lead. So we really want to target layers that are high in uranium and generally low in this common lead. So for example, these layers would be suitable, and some of these here. But if you don't really know where the uranium sits, you can easily do measurements and not produce a good age because you didn't target a layer that has enough uranium for it to be successful. But when it does work, it's great, because what people can do nowadays is collect the same amount of data that I did in 3 years. They can do that in 1 to 2 days. So that's a massive analytical improvement over just a matter of a few years. A downside of doing this is that the precision is quite a bit lower than doing isotope dilution techniques, but it's much faster, so you can produce a lot of data, and if you have information like this and you do need to do solution, then you really can be guided by which sample or which layers do it and need to target. It's also non-destructive, so if you don't have that much material or if you have very precious samples that people don't want to be destroyed, then this is also a very good option because it's non-destructive. Yeah, and I just wanted to show this also, you've seen the evolution of speleothem research in general, but since it was first, sort of the theory published of the potential of doing this, actually there hasn't been that much after that in terms of uranium lead dating. Not even so much after 2006, when it was first, like the real mass spectrometry protocol was published. And then after this nice paper review on what the potential was of this chronometer, it still didn't change that much. I can probably tell you each of these peaks which people published this. But I think, at least in the Speleothem community, people have realized the potential of this, particularly also in like to understand Paleo-Climate further back in time in the terrestrial realm, but also how we can use that information for early human evolution and understand what climatic conditions were then, that there's huge scope to do more of this. And I really hope that this field is starting to pick up, and I do think it's going to do that. So now I'll move to 2 examples of applications of uranium, thorium, and uranium lead dating. So first I'll talk about a record that is just based on uranium, thorium dates, which was published earlier this year by me, led by me and Kil Snyderman and a great team of other people. And in this study, we looked at the Southern Hemisphere subtropics and actually found from these proxy records new data and already published data that the wettest times actually episodically occur during glacial periods, and that the interglacial periods were consistently dry during the Pleistocene, at least over the last 350,000 years. So this challenges what we call the Southern Hemisphere glacial aridity paradigm. And what we mean by this, is that historically this idea developed for a good reason based on evidence, obviously, the idea about what glacial environments and interglacial environments look like. So glacial environments have generally been characterized as harsh and dry, dusty, not that many trees, and interpreted as often being inhospitable for many life forms, where this idea developed that a lot of these life forms, plants, animals, humans, were restricted to these pockets with better conditions called glacial refugia. And on the other hand, interglacial environments were characterized as warm and wet, with widespread growth of trees and probably also widespread habitat availability. But this idea has been largely been developed from evidence from the Northern Hemisphere and a bit biased towards marine and ice core records. Also biased towards the idea that because we see a lot of trees during the interglacials and not much during the glacial times, that this is a sign of that it wasn't wet, but it must have been dry during glacials. But there's 2 aspects that have been often overlooked. So the effect of low glacial atmospheric CO2 on plant growth, particularly what kind of plants can grow, and then reduced evaporation under lower temperatures. So with the first point, we mean that different plants such as C3 and C4 plants have different photosynthetic pathways. And under low atmospheric conditions, C3 plants have reduced photosynthetic rates. So simply ecological competition should favor herbaceous C4 plants over woody C3 plants during these low atmospheric conditions during glacial times. So that also means that perhaps the absence of these C3 plants, which means then low productivity, because there's this absence of trees and similar kinds of C3 plants, that this has maybe been incorrectly interpreted as climatic dryness. And on the other hand, we have the effect of reduced evaporation under lower temperatures. So this is sort of basic physics, where if your climatic moisture availability is precipitation minus evaporation, then under lower temperatures your evaporation will go down and your climatic moisture balance can shift to positive. And that can happen over seasons, but it could also happen over glacial, interglacial cycles. So we looked and focused at the Southern Hemisphere subtropics, which largely sit between 20 and 40 degrees south. Here's a map of that, where you see the climatic moisture index plotted, where the brown colors are negative moisture balance, and blue is positive for the 2 seasons of winter and summer. And we focused on the Southern Hemisphere tropics because they're largely underrepresented in paleoclimatology. And there are records, there are good and interesting records published from these areas, but they are often short or discontinuous or don't have good enough chronology, and sometimes all 3 of those. So while we understand the changes in global temperature quite well, it's not that well constrained what the terrestrial response of these changes in temperature are in the Southern Hemisphere subtropics. So we were motivated by this need for terrestrial records that are really a proxy of moisture and that are independent of vegetation, so that we don't have this problem of C3, C4 plants, and that's extended over multiple glacial, interglacial cycles. I'll just quickly go back. We've looked at 2 new sites from southern Australia, the Lewin Naturaliste caves in the west and then the Nercourt caves in the east. And we also looked at already published records for our Southern Hemisphere comparison, and I'll show you a bit more of that later. But both of these sites are really interesting to understand changes in biodiversity because the Lewin Naturaliste cave sits within the Southwest Australian floristic region. So this is one of the 34 floristic hotspots with a lot of native vascular plants, of which about half of them are endemic. So it's really interesting to understand the climatic conditions, to ask ourselves questions, why is this area so diverse? On the other hand, there's the Nercourt caves in the southeast of Australia, and this is a really cool fossil site with lots of vertebrate fossils, including the extinct Australian megafauna. So in these caves, we have these beautiful chambers decorated with speleothems, but we also have these chambers where you find these large sediment cones that formed underneath these old cave entrances. And it's in these clastic deposits that we often find the fossils. So for anyone who doesn't know what extinct Australian megafauna looks like, it's basically all the cute, funny animals that are there now, but then a lot bigger. So these giant kangaroos up to 3 meters tall, and these really huge herbivores, and even marsupial predators, Tylocoleo. So it's really fascinating to understand why, during this window, did these animals go extinct. So speleothems from drylands are particularly useful because they're so closely linked to the hydroclimate. For them to form, they need at least above 0 degrees temperatures, and they really need a positive moisture balance to be able to form. And they are, the changes then in speleothem growth are really closely linked to changes in the hydroclimate. Where simply put, when you find many speleothems growing at the same time, you will have a lot of water, like drips of water dripping, representing wetter times. Whereas on the other hand, when you have very few or no speleothems growing, this indicates there wasn't so many water drips, and these were times that it was quite dry. So before doing this study that I'll show you, we've also looked at how can we use age data to understand these changes in speleothem growth. So looking at the age distributions from this cave region. But we first wanted to know, if we do that, how many samples do we need and some other factors to consider. So we did an inverse model to understand these kind of limitations to using this technique and how we could do that well and reliably. So I won't go into the detail of the statistics, but if you're interested, then have a look at this paper. But as I said, in short, when we have a lot of speleothems growing are the wetter times versus little speleothem growing are drier times. So the results of that you can see over here. We use a kernel density estimator, which is a continuous probability distribution for the ages. So how we do this is by summing the probabilities of each individual age. And we did Monte Carlo resampling from the underlying age uncertainty of these ages to take into account this varying age uncertainty. And you can see that in the different shadings of the bands. So that's the 1 and 2 sigma bands. Plotted underneath is the age data. So this is for the Nercourt caves, the age distribution, and this for the Lewin Naturaliste caves in the west. And the underlying age data, which is based on over 300 uranium thorium dates, and they all come from individually dated speleothems. So each age represents one speleothem. We did that to not be biased towards a certain age. Also plotted underneath is the Southern Hemisphere summer insolation, and at the bottom the Antarctic temperature stack, just for regional temperature reference. So what this shows is during the Pleistocene, I've plotted these gray bars to highlight the warm periods, and we see that consistently there's minimal growth during the warm interglacials. So it was actually dry during these warm interglacials. The speleothem growth here is precession-paced and often closely following high summer insolation. We then, to understand if we were really looking at proxy of moisture, we also extracted pollen from the same dated speleothems. So this is the pollen we found in these dated speleothems, where the top bits in this sort of greenish color are the climatically informative pollen taxa. And based on these taxa, we did quantitative climatic reconstruction. So we look at the modern distribution of these pollen and then extracted the climatic parameters such as the climatic moisture index to reconstruct the moisture index in the past. So what this shows is that the peaks in speleothem growth nicely align with some of those peaks that we see in climatic moisture. We then zoomed out and looked across the Southern Hemisphere subtropics and pulled out already published records that satisfied our 3 criteria. So we wanted them to be really a proxy of moisture, that they were at least extending up to and including the last interglacial period, and that they had relatively good chronology. So we see the new data from Australia at the top, and then from Southern Africa and Southern South America, and again subtropical Southern Hemisphere summer insolation at the bottom. And if we look at MISE, we see that it's consistently suppressed in all of these records. So also this dry last interglacial period across the Southern Hemisphere subtropics. And most of these records are also precession-paced, closely following Southern Hemisphere summer insolation. We then wanted to look at the latitudinal limits of this pattern. So the pattern that I'm referring to is that during the cooler times it was generally or episodically moist, whereas during the warm periods it was consistently dry. And we did that by comparing these published records, but also pulling out a few more further north and south of the subtropical band to see where, at what latitude this shifts back in the more traditional cool, dry, warm, wet pattern. And we did that by comparing all these proxy records to the Antarctic temperature stack and then extracting at what temperatures peak moisture occurs. So this plot basically sums up what we've seen in the time domain in the previous plot, where the blue indicates locations where peak moisture occurs during temperatures cooler than Holocene, and the brown indicates locations where peak moisture occurred at temperatures higher than Holocene. So this shows that consistently across the Southern Hemisphere subtropics, times of highest moisture availability occurred during temperatures lower than Holocene. So the warmest times sit within the glacials, whereas equatorward and poleward, we see that the times of highest moisture availability occurred during temperatures higher than Holocene, so generally wetter during the warm interglacial times. We couldn't establish it for South America because we didn't have suitable records, and also not for the poleward limit for Africa, but with more suitable records becoming available in the future, we could obviously, hopefully, fill in this plot and determine these latitudinal limits better. So to sum up this bit of the presentation, the implications then for glacial environments are that we should really move away from this binary view of glacial environments, because it's probably a lot more nuanced and might even shift from region to region. And particularly for the Southern Hemisphere subtropics, this shows that glacial periods were actually times that were relatively stable environments with possibly enough or abundant water resources. And we can ask ourselves the questions, were glacial periods really obstacles for the movement and expansion of plants, animals, and humans? Okay, moving on to application of uranium-lead dating. We're staying in the Southern Hemisphere, but zooming in on South Africa to the cradle of humankind. Probably a lot of you are familiar with this beautiful site where, together with Eastern African hominid fossil sites, they really preserve one of the richest fossil records in the world. And yeah, we find these paleo cave remnants, and I say remnants because a lot of these caves have been heavily mined during the 19th century for lime. So a lot of these caves have been blown up and most of the material removed, which is unfortunate for the research, but on the other hand, maybe no one would have ever found the fossils if they wouldn't have mined it there. And fortunately, still enough material is preserved there to be able to look at the fossils and also study archives that are useful for paleoclimatology. So in the cradle of humankind, we find these alternations of speleothems and clastic layers, a little bit analogous to the volcanic tuff layers you find in Eastern Africa, which have been really important to understanding the chronology there. We don't have these volcanic tuff layers in the cradle, but as I said before, speleothems can be dated really well as well. So they've also been very important for the chronology of the cradle of humankind and understanding the ages of the fossils. So this picture nicely shows these alternating layers of flowstones, and then this is FBU, we call this flowstone bounded units. So these are the clastic layers that are then sandwiched between these flowstone layers. So when you date a flowstone at the top of the bottom, you can provide these bracketing ages for the fossils that sit in between in these clastic layers. So there are several techniques that people have applied to understand the chronology at the cradle of humankind, both dating the speleothems and the clastic sediments using, for example, paleo-mag and cosmogenic nuclides. But probably one of the most important chronological information has come from uranium-lead dating of the flowstones. And this was done by Robin Pickering, where she produced a cradle-wide chronology from a lot of samples across the cradle region. So nearly all of the sites are dated except for these three, and our PhD student Georgina Lutti is focusing on dating Motsetsi and two paranthropus sites that don't have absolute ages yet, from Kromdraai and Gondolin. So the chronology is basically well established, but there's hardly been any proxy work done. So that's where my work has come in, and where I focused a lot on developing proxy records for these already dated speleothems. Because we're in the uranium lab world, we'll never be able to produce sort of classical speleothem records with age depth models of the chronology. So we're just looking at sort of snapshots in time, and we have to basically look at the relative variation. But this is still very useful information at the time scale that we're looking at. So I spent this year visiting the Max Planck in Mainz, hosted by Hubert, to do some of these nice proxies that they have available there. So we looked at fluid inclusions and the stable isotopes of the speleothems themselves, and then also some TEX86. So this is some of the results for the fluid inclusions, where here we have the deuterium isotopes and the oxygen isotopes. And this blue line is the global meteoric water line. Plotted in these small dots is the modern rainwater around Pretoria, and with just a linear fit through it. The mean value of the modern day rainwater is this gray pentagon, and then the data from the fluid inclusions, where the color coding is for the age. So very red is old and very blue is young. And we can see, based on the modern data, and we know that the modern rainfall is sourced from the Indian Ocean, and most of the fossil data sits quite closely to the modern day values. So I must also disclaim this is very fresh data, so also the interpretation of this is at very early stages, so please bear with me. But yeah, this indicates that probably the moisture source didn't change that much over time, was relatively stable. We also see this diversion from the global meteoric waterline, and this is probably an artifact due to evaporation. And yeah, we're not entirely sure what the exact effect is of this evaporation. Possibly the isotopes become a little bit heavier towards the late Pleistocene, but yeah, we have to tackle this deviation from the global meteoric waterline first. So just looking at the oxygen isotope data, here we see that for the fluid inclusions, and this for the speleothem calcite itself, and this is from some modern speleothems that we collected, so that are actively growing now. So you can sort of see a line going up a little bit, but we haven't been able to very well constrain this younger end. And unfortunately, I don't have suitable samples to constrain this better, so it is what it is. But for the speleothem stable isotopes, yeah, we also at this stage see a slight increasing trend towards younger samples, and there's scope to fill in this end with more measurements for the stable isotopes to see, do we actually see an increase, or is it basically a flat line? If there's a slight increase, then possibly this indicates a very gradual long-term drying trend. And this might align well with something that we see also in the stratigraphy, where the thickness and the frequency of these flowstones also decrease when we go up in the stratigraphy. Lastly, we looked at these temperature proxies, performing these temperature estimates based on the fluid inclusions, where you combine the oxygen isotopes of the fluid inclusions, so the fossil water trapped within the speleothems, and then the calcite speleothem, or the calcite oxygen isotopes. And you can plot that or chuck that into this equation, isolate T, and then calculate your temperatures. And then using the TEX86, but I won't explain further how that's done because fortunately people have already done that before me. So this is the result we get from the fluid inclusions, which is all over the place, temperatures up to more than 80 degrees Celsius, which is very unrealistic unless we're sitting on top of a hotspot, and we're not. So there's, this is not very useful information, but we can also explain it. So you saw this deviation from the global meteoric waterline. If you then plot the oxygen isotopes against temperature, we see a very strong correlation, and this is just because of this deviation from the global meteoric waterline. Fortunately, we also have measurements from TEX86, and that worked quite well for the first 10 measurements that we've done, and there's more to come. So this is the modern day temperature, and our Holocene measurement, the first one that we have, sits really spot on it's at like 17 degrees Celsius. So that's encouraging for doing some more analysis, and hopefully we'll be able to fill in this more over the last 300 million years. So ultimately we have these different kinds of paleoclimatic proxies that can tell us something about precipitation, the moisture source, and what kind of temperatures we get over the last 3 million years. Maybe seeing a bit of a drying trend, we see that probably this moisture source was stable coming from the Indian Ocean. We might do some triple oxygen isotopes to also be able to say something about relative humidity. We'll get more data from these temperatures, and we're also testing some speleothem microthermometry so that we can independently compare these with the TEX86 temperature measurements. And ultimately, this is just to also address the question, like, what was actually the impact of climate variability on human evolution in South Africa? We see this rapid diversification around 2 million years, and there's faunal turnovers that occur. So it's really interesting to finally have some data, like good paleoclimatic proxy data, that we can address this question with that. Some cheeky promotion before I finish. There's the Karst Record Conference that we're organizing in Cape Town at the end of March next year. There's a lot of speleothem people, but we also have some interesting sessions, and for fossils, archaeology, technique development. We have for the first time an EDI session and an open session. So if you're interested, please check the website, and just note that abstract deadline is on the 1st of November. Yeah, I think that's it. I'll leave you with the nice rolling hills from the Cradle of Humankind and some takeaway messages. And I really want to thank all of these people who've have been crucial to the work and really supported me, and also the people who funded me. And thank you for your attention.